Hi there. So in this scenario, I have a friend who has never had to have a nasal swab throughout the entire pandemic. It's a miracle that they have never had to go through this process. So I'm just going to be explaining kind of the process of what happens when through the entire testing process. So COVID-19 is a highly infectious virus that is transmittable through saliva and mucus droplets of an infected person, and it infects living cells. Virus is highly transmittable and highly contagious. So it is important for someone who's experiencing symptoms of COVID-19 to remain home and to isolate from others so as to reduce the spread of the virus. The principle behind the RT-PCR, so according to the lab materials that were provided for us on this topic, the RT-PCR is a variation of a PCR protocol. It's used to amplify nucleic acid using primers, so DNA primers. COVID-19 is a single strand of RNA, and so there's not enough to work with to fully understand what the genetic material is of COVID-19. And so in order to make a complementary DNA strand, we go through this entire process of reverse transcriptase. So the protocol is a process by which the RNA strand is analyzed and converted into a complementary strand of DNA using DNA primers. Then the reverse transcriptase reads the nucleotides in the RNA and synthesizes a complementary DNA strand. The newly synthesized strand is then amplified exponentially through a PCR reaction. So you have this strand of RNA. And then it goes through the RT-PCR and transcriptase, and it's going to make you a complementary strand of DNA. Well, then you're going to have these little DNA primers that are going to attach to a section on the complementary DNA strand that has been synthesized. And then you're going to have the reverse transcriptase that's going to attach to the synthesized DNA strand. And it's basically just going to copy the code that the DNA primer has put in. So then what happens after that's been done is that synthesized DNA strand that's been completed through the reverse transcriptase is going to split. And then that is going to just have this process continue and continue and continue and just making multiple copies of the synthesized DNA strand. According to the Science Direct website regarding viral targets, it says that in the past, the therapeutic strategy for antimicrobial pathogen primarily targets pathogen genes and proteins. This strategy works well for the antibacterial in most of the cases. Still, it has much less success in antivirus therapy, as the viral genes have an intrinsic nature to mutate very frequently and become resistant to vaccines and drugs. And it goes on to talk about virus replication and all these different things. And so basically what I'm gathering out of this is that there's no real way to have a viral target for a virus. So for a bacteria, it's a lot easier because bacteria don't really do all this weird mutating and stuff. And so with a virus, it's just constantly mutating and constantly replicating and making new strands. And it's really hard to stay on top of a treatment for it because the mutation process is just... once they find a solution to the most recent mutation, it becomes obsolete because then you have this new strand and then basically the process just starts over again. And there's all these different subfamilies and all these different things that come out of this recent mutation. So it's just a really hard process. Interpretation of a positive and negative result, very easy, right? You're positive, you have COVID. You're negative, you don't have COVID, or you're not currently... you don't have the COVID virus active inside of you, and so that's why it's not detecting on your test. So usually wait about 24 to 96 hours after your symptoms have started before testing. And the significance of the test in detecting recent infections or active infections versus past recent infections, obviously it's a new strand, and so it's going to be easier for researchers to take that new infection and hopefully use that to create a treatment for the current strand that they're working with and create a vaccine for it. That's all I have.